Right, afternoon everybody. So I'd like to be able to talk some more. I guess most of you saw what I would say a couple of days ago. Today I'm going to be a little more hardcore and get down to some of the more detailed biology of the stuff that not only we do, but also the stuff that is being done by laboratories and other groups around the world. And a lot of what I want to do is to emphasize how much progress has been made in pushing forward the rejuvenation paradigm, shall we call it, over the past decade or more, and at an accelerated rate. So obviously this is a slide that you've seen a couple of days ago, and those of you who weren't here have probably seen it in about 1 million of the talks that I've given since the dawn of time. Just a couple of minutes there to explain what I mean by it. As I always say, aging can be defined in many ways, but the particular definition that I prefer has the advantage that it demystifies aging. It explains it in terms that do not sound, esoteric and weird, but rather in terms that emphasize how simple a concept aging is. And in particular, that aging is, I like to say, a phenomenon of physics rather than a phenomenon of biology. In other words, it's basically the same thing in living organisms that it is in non-living, man-made machines like cars or airplanes or whatever. Namely, that basically any machine with moving parts, whether we call it alive or not, is certain to do damage to itself as a consequence of its normal operation. And so that's the left-hand arrow down here. Do I have a counter? No, that's bad. That metabolism, which of course is the word that biologists use to denote the entire network of processes that keep us alive from one day to the next, generates changes. And the reason that the word damage is the appropriate word to use to denote those changes, those alterations to the structure and composition of the body at the molecular level, the cellular level, the reason that's the appropriate word is because when you have too much of that damage, you get sick. In other words, the body is set up to tolerate a certain amount of that damage, but only a certain amount. All right, good so far. So the question then is, what are we about here as biomedical gerontologists? In other words, people who want to actually do something about this, rather than simply understand it, which is what the term biogerontology is often used to denote. What we want to do, of course, is separate metabolism from pathology. We want to allow people to continue performing metabolism, in other words, being alive, without getting sick. And without going into too many details, because I know most of you were here a couple of days ago, the essence is that the first approach that people used to try centuries ago was to kind of separate metabolism from pathology by separating damage from pathology. In other words, to attack the health problems of late life in very much the way that one might attack an infection. And then, starting maybe a century ago, a few people started to realize that you couldn't really do that, and that a better approach might be to be more preventative and try to separate metabolism from pathology by separating metabolism from damage. In other words, making the body run really cleanly in a manner that doesn't generate damage, or at least doesn't generate it as rapidly as it naturally does. And that turned out to be a complete non-starter as well, because the creation of damage is just too intrinsically intertwined with the processes that metabolism needs to do to keep us alive. So then, 20-odd years ago, I started to promote the idea that maybe there was a third approach, namely to separate metabolism from pathology by attacking the damage itself, by letting metabolism generate damage at the natural rate, and we haven't got a choice about that to speak of, but by removing that damage periodically to some extent, not perfectly by any means, but to some extent, so that it does not reach the level of abundance that causes us to get sick. And that's all very well. But that idea took quite a while. I'm going to say the best part of a decade for me to really sell to my colleagues within the biology of aging community. And it would have taken a lot longer if I had not been able to flesh it out into a bunch of absolutely, clear-cut, concrete proposals for ways in which the maintenance approach, as I'm calling it, the damage repair approach, could be implemented. And so here is basically what I started saying nearly 20 years ago, actually more than 20 years ago, this actual slide has been on my slide deck for well over a decade. So what I'm actually going to do today is I'm going to spend just a few minutes going through the things on the right-hand side of this slide, the various repair approaches that I was favouring starting 20 odd years ago. And then I'm going to spend most of my time talking about where things have gone since, because, a lot has happened. So first of all, let's talk about the purpose of this way of classifying things. As you will remember, as you were here a couple of days ago, this is kind of the precursor to what are now fashionably called the hallmarks of aging. This was before hallmarks were cool. In fact, it was more than a decade before hallmarks were cool. It's basically a classification of all of the various types of damage that are included in the previous slide, a classification into categories that can then be mapped to ways to repair them. And I feel that, there's lots of discussion about how many hallmarks there are and exactly what they ought to be and everything. And really that discussion, to me, misses the point, because all of the modern, newfangled ways of classifying damage in aging come down to really just classification for the sake of classification, without having this mapping, this one-to-one mapping from the type of damage, the category of damage, to the way in which we might fix it. And I feel that that is really the point. If you don't do that, you're missing the point. So on the left-hand side, you've got these things, cell loss, and what I call division-obsessed cells, which basically means cancer, but also includes things like clonal hematopoiesis, which is quite a hot topic these days. Then death-resistant cells, which include senescent cells, but also include cells that would not be called classically senescent, like, for example, misbehaving immune cells, which are often called anergic. Mitochondrial mutations, which is changes to the sequence of the DNA that we have in our mitochondria, very, very small amount of DNA, but absolutely vital for life. Accumulates mutations much more rapidly than the chromosomes in the nucleus of the cell. Intracellular waste products, junk that accumulates inside cells, and that is initially harmless in very much the same way that your kitchen works perfectly well for a week of accumulating garbage in the trash can, but if you leave it for a month without emptying the trash can, then there's a problem. I have always chosen to distinguish between waste products that accumulate inside the cell and ones that accumulate outside, and that's actually no waste product, of course. And the reason I did is because they had different approaches to how we might get rid of them, which is on the right-hand side, and I'll come back to that in a moment. And my last category number 7, which was unaccountably left out of the original Hallmarks paper in 2013, I ridiculed them quite a bit after that, is the changes to the extracellular matrix, that is this lattice of proteins that that basically gives our organs and tissues their shape. We think about the skeleton and bone that keeps us the shape we are, but the extracellular matrix is just as important, and in particular, it needs to be elastic in certain cases in order to do its job, particularly in the major arteries, and that elasticity is lost progressively over time. So those are the categories that I listed back then, and the good news is that we haven't really needed to update this list, that things have not been discovered in the past 20 years that conspicuously don't fit into any of these categories and require an expansion of the list. So that's handy. It kind of, gives it's standing the test of time. It kind of proves that we are relatively unlikely to find additional types of damage that don't fit into this classification in the future either. All right, so then let's look at the right-hand side of this table. And here what I've done is I've listed one generic approach per category. I said, okay, this is the kind of thing that we could do to fix pretty much any example within the category. And that's an important thing. So I used to talk about types of damage, but I realized recently that the word types is too ambiguous. And I very much distinguish between damage categories, which there's only like 7 of, depending on how you count it, and damage examples, which is, there's hundreds of those, because within each category there are a bunch of examples. So if we look at cell loss, what's that? It's simply cells dying and not being automatically replaced by the division and differentiation of other cells. And of course, that's kind of what stem cell therapy is for, right? You put cells back in and they divide and differentiate to replace the cells that the body is not replacing on its own. Stem cell therapies have other benefits in terms of the stuff that stem cells secrete, but that's for different reasons. If we look at, for example, Parkinson's disease, then undoubtedly the problem is that you lose too many of a particular type of neuron, called a dopaminergic neuron. And the most promising way to address this is indeed to put precursors of dopaminergic neurons into the right place in the brain and let them do their thing. And there's already a clinical trial well underway in Japan doing exactly that. There was actually an attempt to do this 30 years ago when we knew virtually nothing about how to manipulate stem cells in cell culture before injecting them. And even then it was occasionally successful. And when it was successful, it was spectacularly successful. So there's a lot of optimism around that. Then division of cell cells. So as I said, this mostly comes down to cancer. In fact, almost entirely comes down to cancer. And, there's a very fashionable way of saying, it's a thing that people say, like, cancer isn't one disease, it's a whole bunch of different diseases. And of course, that's true by some definitions. Different cancers from different tissues have different characteristics. But the fact is, it's useful to look for things that all cancers have in common, or at least that almost all cancers have in common. And it turns out there is one. There's this thing called telomere maintenance. So telomeres, as I'm sure almost all of you know, are these DNA sequences at the ends of our chromosomes that are very specialized and are used for a variety of purposes, but especially so that the DNA repair machinery in the cell does not simply join chromosomes together end to end. Chromosomes get broken by, radiation and stuff all the time. Maybe you have this thing called a double-strand break, and there's a couple of rather sophisticated systems that cells have to put those breaks back together. But of course, you've got to be able to distinguish. That machinery has to be able to distinguish between what's an actual break and what's the natural end of the chromosome, or else it's going to stitch chromosomes together end to end. That's called a Robertsonian fusion. And, it's very bad for the cell if that happens. So, telomeres are necessary. It turns out, because of the way that DNA replication evolved, that telomeres get shorter when cells divide, and also for other reasons, but especially when cells divide. And as a result, the telomere will eventually disappear, and you'll get this thing of chromosome fusion. Very bad. So evolution knows about this, of course, and has fixed the problem by developing a system called telomerase, which basically repairs the ends of the chromosomes, re-extends them to compensate for the loss of DNA that happens during cell division. And a division-obsessed cell, which is dividing a lot, needs that system. Now, evolution knows that as well, and the result is that organisms like humans simply don't make this thing telomerase, or at least they make it in certain cell types, but they make as little of it as they can possibly get away with. And the reason they do that is so that it's difficult for cells to mutate into a state where they make a lot of this telomerase thing and thereby can divide indefinitely. However, that's exactly what happens. 90% of our cancers express this one enzyme. The other 10% express something that works in a different way but still has the same consequence of maintaining telomeres. And so, it might be good to actually start to stop that from happening. And 20 years ago, I decided that, that was probably the most promising generic approach to actually hitting cancer on the head. Then death-resistant cells. So as I said, that more or less means senescent cells. So there are some other examples that would not be classically called senescent. And back in the day, I was thinking, well, this is pretty tricky to do, but there's this wonderful thing that is used in the lab, not in the clinic, but in the lab, called suicide gene therapy. which is a trick where you basically do regular gene therapy. You have a virus which you put in some cargo in, some gene, right? And you get that gene into cells. But what you do is you arrange that the gene is only going to be expressed, in other words, protein is only going to be synthesized from the gene in certain particular circumstances. And in particular, you arrange that the circumstances in question are when the cell has become one that you want to get rid of, that you want to die. And somehow it's resisting the efforts of the body to kill it off. You kill it off by this artificial means. And you do this by basically making sure that the gene that you are expressing in this circumstance is a very toxic one. So that the cell basically dies when it starts making the thing. That's called suicide gene therapy, and even back 20 years ago, it was a pretty routine technique in mice, for messing around with this or that. But a very, high-risk thing to do in humans. But I figured that, in due course, we'll figure out how to do it safely, and so that's going to be the technique. Then mitochondrial mutations. So I mentioned that mitochondria have this very small but very essential genome. It expresses just 13 proteins, but those proteins are absolutely essential to our ability to metabolize oxygen, in other words, to breathe. And the thing is that the mitochondria itself is a really, really complicated machine that has far more than 13 proteins in it. It has well over 1,000 proteins. And all the others are encoded in genes that are sitting in our nucleus. And so you may ask, well, that's the wrong place, right? But that's okay, because the cell has this clever system for importing proteins into the mitochondria after they've been synthesized in the main body of the cell, the cytosol. 25 years ago or more, I had this bright idea, which was by no means original with me. A bunch of other people had thought of it independently, of saying, well, okay, we've got all these 1,000 proteins that are encoded in nuclear gene. Why don't we just piggyback on that machinery and put backup copies of the 13 proteins that are encoded in mitochondrial genes into the nucleus? And of course, whenever you have a bright idea in biology and biotechnology, you have to ask yourself, well, would evolution have done it if it were that easy? And the answer is usually yes. So of course, one knows immediately that it's not that easy, but it may not be impossible. Evolution is cleverer than you are. That's a quote from Francis Crick. But it also has fewer tools. It only does things by very small incremental steps where we can do bigger things. So this seemed to be worth trying, and people did try it in the 90s. And when I kind of just rediscovered this whole idea and people had more or less given up, I said to myself, well, I think people have given up a bit too easily. So started a project on that back in the mid-2000s. We've been doing it ever since. We've made a lot of progress. Still some way to go, though, even though the progress that we have made is far more than anyone would have expected to be possible. Then there's, as I said, garbage inside the cell. So why does garbage accumulate in the cell? Well, I mean, byproducts of cellular processes of metabolism are created all the time. And, you've got to get rid of your garbage. You've just got to decide. So, of course, evolution, again, knows that. And what's happened is that evolution has developed ways to get rid of garbage, either to excrete it or to destroy it. But unfortunately, evolution only does that when the type of garbage in question is one that accumulates reasonably rapidly. If you've got a type of garbage that accumulates really, really, really slowly, so that it doesn't actually overwhelm the cell until late life, until old age, evolution can't be bothered to figure out a way to do anything about that, because evolution doesn't care about old people or old animals. It only cares about you until you have reproduced. So we have to augment the garbage disposal machinery that the cell naturally had. And I realized a long time ago that we could probably do that using enzymes that we find in other species, especially in microbes. Then for extracellular waste products, it turns out to be rather easier because the machinery that we naturally have outside the cell to destroy shit is far more primitive than what we have inside the cell. So all we need to do is engulf the stuff, get it from the outside of the cell to the inside. And that basically can be done by vaccination. You can actually just trick the immune system into thinking that the stuff is foreign and get it inside the cell and it's toast. And then finally, there's this stiffening I talked about. Turns out that the chemical basis for a lot of that stiffening is quite well known, And so basically, in principle, one can apply chemistry to actually look and develop drugs that react with the chemical bonds that have formed that have reduced the elasticity of the matrix and break those bonds, thus restoring the elasticity. All right, so that's all very well. And as I say, these are ideas that I was already talking about 20 odd years ago, but they've all made progress. I mean, quite a lot of progress. And I think really the best measure of it is what I've written at the bottom of this slide, that this is no longer just some academic or non-profit pursuit, even the most difficult things. So I would say those are mitochondrial mutations and extracellular matrix stiffening. Even they are now being pursued in startup. I mean, actually, I can only think of one startup for each of those two. But certainly, you know, it's a lot better than it was. It's very good news. Things are moving forward. And but I'm going to spend pretty much all the rest of my time on there. I'm going to talk about the alternative fixes. So I mentioned that what we're looking for here is repair strategies that are generic, that can, in principle, be used, perhaps with differences in details, but only details, across all of the examples within a given category, or at least almost all of them. And the thing is, you might have more than one such approach, and that would be wonderful because we don't know which one's going to work best, it's going to be easiest to develop until we get there. So I'm all for diversity of approaches. So let's go through these things. You know, this is really, really good news. With every single one of them, as you can see, it's now also this, now also that. So I'm going to go through them and discuss them a little bit because these are things, some of which you've probably not, almost certainly not heard of. And it's a really good, you know, top-level measure of the rate at which things are moving forward, and it's a kind of, I like to call it a justification for my progressive reduction in my predictions of time frames to get to longevity escape velocity. So first of all, let's look at cell loss. Originally, a moment ago, I was talking about stem cell therapy, putting cells into the body that you have prepared outside the body in the lab into the right kind of state so that when you inject them, they know what to do to divide and differentiate to replace the cells that the body's not replacing like that. But of course, in principle, you could have some intervention that acts on cells that are already in the body that are kind of similar to, but not exactly what is needed to do this division and differentiation. And you can kind of tweak those cells, you know, nudge them into the state that you want some cells in the body to be in. It doesn't actually matter whether you took the cells from outside the body or whether the cells were already in there, so long as they are, after the thing you do, they're in there and they're doing the right thing. And of course, that's exactly what partial reprogramming is all about. We've heard about that from a number of talks over the past few days. We'll hear about more in the other session that Max Humphrey is organizing in a couple of days' time. But of course, this is a system for essentially using the same machinery that the fertilized egg uses to start the whole developmental program, starting from two very differentiated genomes, the sperm genome and the unfertilized oocyte genome. So that machinery can be used, it seems, in a very versatile manner to make cells more stem-like. If you do it in a really heavy-duty way in cell culture, then you can make them very stem-like indeed, essentially similar to cells that exist in an embryo that's 25 days old or so. But if you do it just if you titrate that and do it just a little bit, you can achieve, it seems, pretty much the same thing that we want to do, namely give us more stem cells by stemifying cells that are no longer quite stem cells naturally. Then for division of test cells, so the idea that we could activate the immune system against cancer has been around for a very long time and for a very good reason, namely that it already exists. The body definitely uses the immune system to minimize our risk of cancer in a very powerful way, and it's been well known that happens for a long time. But of course, it doesn't quite completely succeed, or else we wouldn't get cancer. So what's going on there? Well, the answer, of course, is that it's kind of arms race, that the cancer is kind of trying to get cleverer and mutate its way out of any of the challenges that either the doctor or the body itself is throwing at it, and cancers are pretty good at that. So sometimes cancers outsmart the immune system by a variety of methods. And this is why cancer immunotherapy as a field was absolutely unsuccessful. It got more or less exactly nowhere for decades. But then about 10 or 12 years ago, that all changed in a massive way with the discovery of a couple of ways to make the immune system better at attacking cancers. One of those methods has already got the Nobel Prize, and it certainly moved forward very well in terms of commercialization and clinical trials and everything. There's a long way to go to make it work really well, but it's definitely happening. And there are companies in our space that are very interested in doing this, not least because they, like me, regard cancer as very much part of aging, and that are, creating a kind of portfolio of things that, first of all, make the immune system stronger and more, energetic and aggressive against cancers, but also that make the cancers weaker. In other words, make them less able to defend themselves against the immune system. Then this little thing called 6-thiodG is a molecule developed by some friends of mine in Texas about a decade ago, and it finally found its way through as far as phase 2 clinical trials. It's a molecule that basically messes up cells that are making telomerase. You could think of it as, essentially, I mentioned suicide gene therapy a moment ago. This is a molecule that turns telomerase into a suicide gene. In other words, if a cell is already making a bunch of telomerase, then it will kill itself if this molecule is present in the cell. And that's very handy indeed. It seems to work really well. Of course, you may be thinking, hang on, there are some non-cancer cells that express telomerase, and you're quite right, but luckily they express a teeny tiny fraction as much as cancer cells do, so it's very easy to find a therapeutic window there at dose and duration of the treatment that is basically harmless to such cells, but still knocks the cancer on the head really hard. Death-resistant cells, of course, is the biggest good surprise, pleasant surprise that's happened in the past 20 years of the biology of aging. It is the discovery that, I need to do all this complicated nonsense with suicide gene therapy. We can actually have small molecules that fairly selectively knock out senescent cells without knocking out much else. Now, I say fairly selectively, and I will be honest with you, don't really believe anything you read in the academic literature about senolytics, because there's a big reproducibility problem there. Nevertheless, some things are looking like pretty well characterized right now. Those of you who are watching my talk the day before yesterday will probably remember that one of the 4 interventions we are using in mice is a senolytic, which is a pretty well validated one. So but this was completely out of the blue about 12 years ago or so when this started coming out, probably more like 10 years ago. The idea that a small molecule could selectively kill these cells was really, I'd never considered the possibility. Then with mitochondrial mutations, so it's incredibly difficult to get these genes to work when they start out in the wrong place in the nucleus. Quite apart from the fact that you have to do, of course, gene therapy to get these backup copies into the nucleus in the first place. But turns out that cells that are affected by mitochondrial mutations still have some normal, non-mutant mitochondria. And so if we could somehow, reverse the selective pressure that exists and get the mutant mitochondria to be depleted by being outcompeted by the non-mutant ones, then that would be great. And so I'm mentioning a couple of approaches here. There's a protein which is part of ATP synthase that appears to have some great promise here. If you can inhibit its activity, then it may actually make mutant mitochondria more susceptible to the process of destruction called mitophagy that people have been studying for quite a while. And that's an idea that I'm very interested in. And then there's another thing which is a bit more technical, but basically it involves creating not copies of the genes that you put in the nucleus, but rather engineered versions of the mitochondrial DNA itself that will be engineered to have a competitive advantage relative to the natural mitochondrial DNA. That's the concept called gene drive, which you may have heard of in very different contexts. It's being used to get rid of mosquitoes in Nantucket and things like that, but this is a possible way of going about this. Then for junk, so we've got to get rid of the junk, right? But that doesn't necessarily mean we have to destroy it with enzymes that we find in other species. But that approach is, as I say, quite successful, and indeed there's a company or two that are doing it that way. What it instead do is pull the junk out of the cell and excrete it. That too is something that only happens, sometimes, incompletely. We could do it better. So that's an approach that has been demonstrated quite well in the case of atherosclerosis, where the junk in question consists of oxidized derivatives of cholesterol. And that has led to a company, one of the spin-outs from Health Research Foundation, that's going quite well. Then I also want to talk about a project that's actually going on still within Health Research Foundation, but which I inspired when I was there, and which I believe is very promising, and that is to use a special type of antibody to attack the garbage in question, in the case where the garbage is made of proteins. Before telling you more about that, I'm going to go on to the next one, because the next one is using the same kind of technology, but in a slightly less sophisticated way. With garbage outside the cell, as I say, if you can get it inside the cell, then great, but that's not as easy as it sounds. People have been trying to do this in the case of the garbage called senile plaques in Alzheimer's disease for more than 20 years now, and it's, to those of you who follow the biotech world, it's been a very rocky road. There are some things that seem to do that. They don't have much clinical benefit. It's like a bit dodgy. But the point is, you can't get rid of this stuff that way sometimes. But another way to do it is to chop the garbage up in situ rather than getting it inside the cell. And that's what these things do, these catabolies. So catabolies are a type of antibody. So antibodies normally, of course, they have an antigen. They have a cognate thing that they bind to. But catabolies are a special type of antibody that doesn't just bind to the thing that it's cognate for, it actually chops it up. It's proteolytic if the thing in question is a protein. And catabolies are, again, some work that we found in the Health Research Foundation that was spun out into a company called Covalent Biosciences is using that to get rid of certain types of extracellular junk that accumulate in the cell. So this is a very exciting way to go, and it definitely augments what we can do with the immunotherapy approach to engulf the stuff and get it inside the cell. So going back now to cytosolic catabolies, the thing about antibodies is they are outside the cell, they're in the bloodstream. But not long ago, 5 years ago, a method was discovered for getting antibodies into the cell, going through the cell membrane and into the main body of the cell, the cytosol. Now that's really important because it turns out that the cases that we know about, the prominent cases where the junk inside the cell is made of protein, that junk is in a different part of the cell than your non-proteinaceous junk. Your non-proteinaceous junk is usually in a particular compartment called the lysosome. And that's kind of a good thing because it's out of the way, it's sequestered. And similarly, if you put some foreign enzyme into the cell and you want to chop this, want to get rid of this stuff, that foreign enzyme also has to be in the lysosome. And that's good because, cells get unhappy when they see foreign enzymes. But catabolies are antibodies, so it's not the same thing doesn't really apply. And now that we can get them into the cytosol, the main body of the cell, we can use them to attack these proteins. It's very important to use something like an antibody because antibodies are very, very specific, of course. A particular antibody will only bind to a particular protein. It won't bind to any old protein, and that's a damn good thing because we don't want to chop up all our proteins. We want to chop up only a particular one. All right, so the final one is ECM stiffening, so matrix stiffening. So I mentioned earlier that there is an approach to restoring elasticity of the ECM that involves chemistry, involves breaking cross-links that spontaneously accumulate and that reduce the elasticity. And that's very important, and as I mentioned, there's a company, again, spin out from Health Research Foundation that is working on that. It's called Revol Pharmaceuticals. That's great, but another approach that has great promise is simply to turn over, to recycle the ECM. Of course, at a modest rate, so that you don't end up, collapsing in the meantime, but still to continuously or periodically just break down some of the ECM and rebuild it. And of course, you're going to have to ask, why isn't that happening anywhere? And it's the usual reason. Evolution doesn't care about old people. And it turns out, therefore, that it's not actually at all easy to make this happen. But a group at the University in Virginia figured out a good way to do this, especially in relation to possibly the most important protein in the arteries in this regard, namely elastin, which undergoes not only chemical modifications but also physical ones, actual rupture. You can't fix that with chemistry. So this turnover thing is also another very cool idea. So basically, I think I've only got a couple more minutes, so I'm more or less going to stop there. But I was just kind of making sure that you understand that this field is going bloody well. You know, that we are really seeing a huge avalanche, a torrent of new ideas that work, at least in the laboratory, and therefore some of them will, in due course, work in the clinic. And, you know, it's, every time that something like this comes along, it's a new reinforcement of the idea that damage repair, comprehensive damage repair, is really a feasible thing. So I talked about the main project, the flagship project of LEV Foundation last time, and I'll probably be able to talk about it again at the next session a few days from now, so I won't spend any time on it right now. But as I mentioned a couple of days ago, it's the most exciting and challenging and indeed terrifying project that I've ever led. So I'm really, really happy about it. I just thought I would start by, by putting this slide in that I forgot to put in 2 days ago. I was very stupid. So this is a picture from the conference that I ran in Dublin in September of last year, which was a huge release to me. Conferences are a very big deal for me. They are the core of everything I do in community building. And I've been doing them 20 years. They have, you know, had a pretty damn good hit rate in bringing people into the field, creating alliances and generally motivating and mobilizing people to make an impact in the field. So the lockdown was a absolute fucking disaster for me. It meant that I couldn't do one of my favorite things. But of course, we're back now. So last September, we had this fantastic meeting. Up here on stage, on either side of me, you can see the main people who started out as the leaders of our various projects, and also the people who were on our board of directors. But of course, it's going to happen again. Way back in the 2000s, when I started doing conferences, there was, progress was not fast enough to justify a conference every year, only every 2 years. But you've definitely got justification for a conference every year now. And so, of course, we are doing it again. We're going to Dublin, August 17th to 20th. And I gather there's this newfangled thing called a QR code that one can take a picture of and go to the website without even having to type it in. So I think I will just stop there. And maybe there's a couple of minutes for questions if there are any. Go ahead, shout.
Why is the internal data side get problem? And you just kill cells that really.
Okay, great question. So the question is, why are mitochondrial mutations a problem? Okay, so you're not really asking why are they a problem. You're asking, why do we have to use these terribly elaborate ways to get rid of them when, of course, we could just kill the cell that contains them and then we get rid of them by default. Sounds cool, doesn't it? The main answer to your question is 2 words: skeletal muscle. Skeletal muscle accumulates a lot of these mutations, but it doesn't accumulate mutations uniformly. What happens is that, and of course, a skeletal muscle fiber is, a very long cell, right, formed by the fusion of a lot of cells in a line. Now, what happens is that you have this phenomenon called clonal expansion. Actual mutational events in the mitochondrial DNA are not all that common. But the problem is that certain mutations, in fact probably most mutations, certainly most really bad ones, somehow enjoy a selective advantage in the cell. So there's a process of continuous turnover of mitochondria, of course, and it seems that mutant mitochondria kind of stay out of the way of that turnover process. So they proliferate. And maybe some people think they also divide faster. But either or both of those things would have the effects, right? So they clonally expand. But if you're starting with one mutation, one mitochondria that's messed up, then the clonal expansion, just spreads out from there along the fiber. And it turns out that because mitochondria are wrapped around sarcomeres and they're really very immobile in muscle fibers, that this happens really, really slowly. So even in late life, you see, a maximum of maybe 2 millimeter long segments that are completely taken over by these mutant mitochondria. Now, kill a cell like that, what's going to happen? If you kill the entire muscle fiber, bang, you've lost a lot of stuff that's perfectly good. And the problem is, if you do the numbers, that at least a quarter, maybe even most muscle fibers have some segment somewhere in them that's mutant. So you're totally fucking yourself up. You're going to accelerate sarcopenia if you do it that way. I think you can't really get away with that. Yep.
If I heard you correctly, you talked about inserting antibodies inside the cells.
Right.
Can you please break how we propose to put the cells, how they're supposed to work inside, how do you plan to get them out if when they're over here?
Well, so first of all, I'll answer the last part. You don't need to get them out. They get broken down, same as any other protein. So they're not, that's a spontaneous thing that we don't have to do ourselves. But how do we get them in? Great question. Because the natural way in which things can be, induced to go into cells when they're normally outside is a process called endocytosis, where essentially the cell forms this kind of invagination in its membrane, and the thing is in there, and the invagination kind of chips, kind of nips off, and so you've got the thing inside the cell. But that's no good, because what you've got is your antibodies inside the cell, but it's also inside this membrane, and it can't get access to the stuff that we want it to get access to, so that doesn't work. So basically, the reason why this whole idea flies is because of a discovery about 5 years ago, published 5 years ago, that shows how to get the antibodies directly through the membrane, not by endocytosis. So they actually get into the cytosol proper. And essentially it works by attaching a nucleic acid to the antibody. Goodness only knows why that worked. I don't think that's really much understanding of that, but it really does work pretty well. So yes, there is a method to do it.
And how do they work?
Oh, well, I mean, once the antibody's in, it's an antibody, right? So it looks to or binds to its cognate antigen, which in this case might be, for example, hyperphosphorylated tau. And because it's a catibody, it chopped it up. How the catibodies work, I have to send you half a dozen papers, but that's a fairly established piece of biology now. Okay. All right, I think that's it. I'll let Alex do her thing.
Okay. So next lesson will be on where are we with cell therapy, exosome therapy, and I will skip the senescence part because it's way too much, and you already heard about senescence, and we'll hear more about senescence. So I will be talking about where this field of stem cell therapy is, and I put the stem in brackets because it's mostly on stem cell, but there's a little bit of work on cell therapies as well. So let's step back and look at stem cells. So stem cells are thought about the bit of our body that is kind of like relatively stable, relatively what people like to call immortal. But what is really only immortal is the germline stem cells. So that's kind of the green line, and that's kind of like what keeps our species alive. And a little bit below it is something that we call like a pluripotent stem cell. That's, for example, the embryonic stem cells. There are also some publications out that say that the human body also have pluripotent stem cells within the different tissue. It's still a field that is developing. And then below it, you have your normal cells that just divide either until they are senescent or they're becoming cancer cells. And then, you might want to get rid of them. So that's kind of like what we are built of. So a little bit of nomenclature, because this is kind of chaos, and biology didn't do a really good systematic job at making up names. So they kind of like were kind of made up, and like, so it's kind of a bit confusing for someone coming into the field. So a stem cell is basically a cell that can divide, make more of itself, but also has the ability to differentiate into a thing, like a tissue type. And then below that, with a little bit of like less capacity, is a progenitor type. So usually it can do less different cell types, and it also has less capacity to kind of like make more of itself. Embryonic, so that name just basically gives you the information where that stem cell is coming from. It could also be fetal, it could be neonatal. So all of that just are names of like where is that stem cell coming from. Also, you should know the kind of the adult stem cell. That's usually something that we take out of the body. So you might know it from like the cancer field where bone marrow transplantations are happening. Part of the bone marrow are hematopoietic stem cells. And then we have more names. So autologous, allogeneic. So autologous is something that comes from myself and goes back into my own body. So that's basically cell therapies or stem cell therapies where you're donating your own cells, something is done to it, and then it goes back into your body. Or you have allogeneic, that's something else, someone else donated, or it is some artificial form of stem cells. And then, yes, people have started editing these cells and kind of like playing with them. So now you have also what we call a cell and gene therapy product. So that's enough. You can read it later at all like your leisure. So how does it look in the body? So with the hematopoietic stem cell, how do we make all the different blood types in our cell, in our body? So this was kind of like an old model. Again, it will be out there. You can look at it in leisure. But recent progress in single cell sequencing basically helps us to better understand how that hierarchy works and how can we push into a certain direction if we, for example, are missing one particular stem cell type or immune type. Maybe we want to have a T cell therapy. So we want to make T cells. How do we push the cells into that direction? So this kind of hierarchy is very informative and helpful for us. But unfortunately, instead of becoming easy, it's becoming more complex and a little bit more vague. So now we're learning that the thing up on the top, the hematopoietic stem cells, is actually not one little identity of a cell. It's kind of more a thing that lives in a space, it's less defined, and there might be some natural reprogramming happening, like in the body. There's some flexibility within the cell state. But we do learn way more like which markers they express, so we have better kind of tools available to identify them, manipulate them, and make them better. But I think it's still a model that is very much evolving.
Right.
So when did stem cell therapy basically start? It has been around since a while. It started basically from the transfusion medicine, basically where like first blood, and then later bone marrow has been transplanted. It's relatively easy on like a small scale, but becomes more kind of like cost intensive if you go large scale. Since the 1990s, it has been more linked to kind of like aging, but unfortunately we don't really have great success yet in the field. It's getting slightly better with the development of CAR T cells against cancer cells, kind of like we have some success, but we have many failed trials. So regenerative medicine was super hyped. Everything thought that's the way how to do it. That's the way how to replace kind of like your failed cells in the body. And unfortunately, we also have a lot of kind of like people that just do cell therapy. They call it cell therapy. It's very hard to know what kind of thing they have. It's not regulated. So it's also burdening the field a little bit. I don't want you to judge that too harshly because it's also, FDA is restricting very much what we do. But the problem is there's kind of no openness of like what the product is. Usually they just say, we have a stem cell therapy. And then you as a consumer don't really know what you will get injected with. Stem cells, so they're kind of like a model there, and you wouldn't know, unfortunately, there's tons of things happening to the stem cell while we age. And basically, these are just kind of like single examples that you can look at in more detail. They age in the same way that any kind of other cell type in the body ages. They have a little bit more resistance against kind of aging, so they have a little bit more DNA repair capacity. They are a little bit more resistant to oxidative stress. It might have slightly longer telomeres, and they have a little bit of telomerase activity, but in the end, they all age. Is that true? Is the human body actually ever running out of stem cells? And yes, there are some publications where people, like the top is on hematopoietic stem cells making your blood cells, that eventually, yes, we're running low on hematopoietic stem cells making our blood. So if we're living longer, eventually that will give us the problem of like, we actually don't have any HSCs left that can make blood. On the bottom is actually how many neuronal stem cells are found in the brain, human brain, and basically that's even more dire because with 2 years, you're out of stem cells. There is a lot of contradiction in this field, like people debating, do progenitor stem cells in the brain exist, yes or no? A lot of the data comes from mouse, and mouse seems to be a very bad model at predicting what happens in humans. So the field is still kind of debating it, but mostly we're running out of stem cells in the brain pretty early. How is it linked to lifespan? Is there kind of like, can we do something about it? So if we're running out of stem cell, can we not simply replace it? And there's like one publication here on where in mice they did some transplantation of young MSCs, and yes, the lifespan is a little bit pushed to kind of like increase, and that's great. So fantastic, it works. It works also for a different cell type here, for the hematopoietic stem cells. Again, just a young to old transplantation helps the mice to live longer. Unfortunately, the quality of the studies, as I have said earlier, is kind of like debatable. The numbers of mice in the groups are kind of low. Often they're using, like here at the bottom left, a very strange kind of accelerated aging model. So how much that tells us about like normal aging is questionable. So kind of the field is not well developed. We need tons more. It is, but what's happening in humans? So that's all mice, it's boring anyway. What's happening in human? And if you want to really kind of get a snapshot, you want to dive into this, here are all kind of like registers that are online that you can visit that's updated when a clinical trial is kind of done. And I looked at what aging-related trials I could find, and the closest I could get to is frailty. So there are actually 13 running clinical trials on frailty, one in China, one in Japan, and 2 were kind of registered in the World Health Data Bank. What else? So it was frailty and skin aging. Very limited scope, but at least there are people trying it. And here's kind of just one example. They have now published some of the phase 1 study data, and now phase 2 is running. It's from a company, Langbarun. Look them up. It's just kind of like one snapshot, and simply because this seems to be the company that is most active in the field, they have the most clinical trials running. And they had, in the phase 130 subjects, they tried to do a little bit of dosing to see how many cells do we actually need. And then the output, the primary endpoint is a 6-minute walk test that we have already heard earlier as like, that's a kind of a very classical biomarker of aging. And then as secondary endpoints, they are kind of frailty status, inflammation, quality of life. And they also now have the phase 2 data out, and it seems to look very positive. So watch out, they probably will have a phase 3 soon. But yeah, so I looked at the FDA and I tried to find kind of like what's out there. And unfortunately, I only found like 2 products that are approved. So basically, that's things that you can get. There's fibroblasts, not stem cells, fibroblasts that you can get against age wrinkles. And there is some chondrocytes that you can get if you have damaged cartilage, which, yes, is very much associated with aging. And then the rest is all cancer. So not a lot of products yet approved. So question is then, why are we so bad? Why are there so many kind of failing in this industry? And here is just kind of a list of roadblocks that I think I came across that make sense. There might be tons more, but we are pretty bad at getting so in graftland, really getting the cells back into the niche and have them there sit and do their thing. It's pretty difficult, and we do seem to not fully understand how we can manipulate them. If we age, our stem cell niche is also aging, so there might be further hurdles if you're working with elderly people. Cost, I had like earlier some modeling on like what cell production actually costs. So cost, unfortunately, is very much a problem. We simply need better ways of engineering. We need better kind of media that cost less. And then We probably also need better cryopreservation of the cells. We don't do anything about senescence. I've seen kind of like expansion protocols that push cells to the limit. So how much these cells are actually still functional is often not tested. So you might get a cell product injected, but how functional that is often not understood and often not tested deeply enough. Immune rejection is a problem. It's a hot topic. So there are a lot of people working on like how can you maybe have like an iPS cell line that is universal usable, so that isn't recognized by the immune system, and I could use it for everyone in the room. So that's great. If you look at like how many lifespan studies there have been preclinical, not a lot. If you look how many more have been done on drugs, way more. So there have been 102 drug-related lifespan study, and roughly about 15 that are related more or less to the cell therapy field. And then the last, I think the greatest problem is we don't really know how they are working. And if we don't understand the mode of action, it's difficult to kind of like improve what we're currently doing. So how do they actually work? So initially we thought, okay, we just replace a cell. So I'm missing a mesenchymal stem cell in my bone marrow, so I just put like a new one in. But we learned often our stem cells just are around for like a short while. So maybe it's not really replacement. In some cases for like hematopoietic stem cell, that's true. But for other cell types, they might do other things. They might be modulating your immune system. So kind of like that's kind of the question. Sometimes tissue integration is really hard. So people with muscle frailty, getting a stem cell into the muscle, super hard. They don't want to go there. They have to migrate there. And then they have to fuse with that muscle fiber. Super hard. So we have to kind of learn to guide stem cells to the places that they need to be. Immune rejection is a problem. Cells are producing tons of factor, so the paracrine secretion of them. We are starting to understand that better with kind of metabolomic, proteomic studies. We're trying to understand, okay, so this cell is actually producing these factors, and it's actually the factors themselves that do something to the surrounding tissue. Mitochondrial transfer is happening, so some people claim that like the cells that we're transplanting, what they do is actually kind of donating the healthy young mitochondria to the surrounding tissue, and that is what is rejuvenating them. Or it could be that the stem cells you're replacing, they have better enzymes, they're more and better at making extracellular matrix, and part of your old matrix is being replaced. Cells are producing exosomes, so that is something that I want to spend some time on, because that's currently the hot topic in the cell therapy field. So a lot of the companies that initially work with stem cells now learn that, okay, so these cells are spitting out things that might be the really active ingredients. So what are exosomes? They're little bubbles of membrane with cytoplasma in it. And because the field is relatively young, they're still struggling with the nomenclature. So you might hear exosomes, you might hear extracellular vesicles, you might hear other names. Usually exosomes are extracellular vesicle of a certain size, but again, it's not consistently used, and the most sexy term is exosome. So that's usually what you will be seeing even if the product might be an extracellular vesicle. You might have other things in this kind of exosome than cytoplasma, and here's kind of a little bit of a scale so that you get some idea of like other sizes of other molecules. So they are pretty small. They can be of the size of an apoptotic body, but they can also be as small as a kind of a virus. And again, like we're still discovering this field. It's still a lot unknowns here. But how are they made by the cell? So they seem to be kind of like different ways of how the cell is producing that. So it can either be by blebbing, so just a little bit of a membrane is kind of like extracted and becomes its own entity. And it seems also that a lot of this is, again, changing with aging. So again, I won't go through all of the list. A lot of the things are changing. But how are they used? A lot of the use is kind of on the biomarker side. So there is kind of, again, that's a hot topic. They're being used as biomarker of cancer, biomarker of frailty. I think we have to wait really how well they work. And then the other thing is on the therapy side. So basically exosomes are harvested, injected instead of your cell therapy, and then they do something. Or what is kind of, I think, the most exciting bit is where like engineering and biology come together. There's a lot where like drug delivery systems are being created because these exosomes can be filled with your drug of choice. So basically what they are doing is they're just taking the drug, push it into the exosome, and the exosome is an entity that is more resistant to degradation. It's also naturally fusing to your cell membrane, so the delivery is slightly better. And you can make it even more better by, for example, adding a receptor target so that you can make it specific. So you want your exosomes only go to the lung? Sure, no problem. You're adding a protein to the membrane, and then your cells and your drug will only go to the lung. Great. A lot of things going there. And then you can also do vaccines, but then again, that is a smaller field that is not yet well developed. What's currently in the clinic, this is just a figure that looks a little bit like what the usage is. And as I said, like most of the people, 50% of the field is kind of looking at exosomes as a biomarker, but there's a considerable amount of exosome therapy happening. What are the kind of like trajectory? So ignore the conditioned medium, it's going up. So it's kind of a huge trend, and I think a growing market there. Companies in the space, again, for your leisure to look at, it's exploding. Really also a lot of big companies are buying in exosome technologies and exosome therapy. So it's kind of a thing that seems to be exciting, probably because of the engineering aspect, because you can make your product and you can kind of like design it in a way that it becomes more specific than the natural product. Again, as I said, a lot has been done in kind of like pre-clinic. Again, only mouse model, but exciting. They seem to help against Alzheimer, osteoporosis, atherosclerosis, type 2 diabetes, osteoarthritis, frailty. So basically, a little bit of a hype. So I want to kind of be a bit careful and not say this is kind of like the solution, because that slide with the word stem cell was flying around conference halls like 10 years ago. So yes, it's looking promising, but we have to see how that translates in the clinic. Again, have someone kind of like put it into a mouse? Have we shown that there like is lifespan extension? Yes, there's one study published already in 2019 that shows a pretty considerable expansion of the maximum lifespan. So 50% is not bad. Like there are not many drugs that are better than this. So looks good. And they also are trying to figure out how it's working. So they think it's related to NAD, it's related to kind of like delivering more energy to the mouse. There's also more human data coming around, people looking at like if there's association with aging. It's a little bit a subcategory of maybe your blood-based proteomic or metabolomic clocks that you have seen earlier today. So this will become part of this multi-clock activity, I will guess. But not everything is good about them. So unfortunately, also other cells than stem cells are spitting out exosomes. And your favorite senescent cells are doing the same. Your cancer cells are doing the same. Your inflamed immune cells are doing it. So there's a lot of kind of like in the blood that is an exosome. And what we currently can't do is can't distinguish the good from the bad. So we need to develop tools to kind of like recognize them and isolate the ones that are useful, learn what's inside, and then isolate the bad ones. And probably you will have a therapy that kind of get rid of all the cancer-related and senescence-related exosomes and supplements you some good young ones. So, so there's kind of like one example where they actually looked at human frailty subjects and looked at like how much inflammatory proteins are in these extracellular vesicles, and they find that in frail elderly people, you have more of these kind of bad molecules. So if you would just simply go and harvest it from an old person and put it to another old person, you would basically do harm. You would kind of transfer some proteins that are pro-inflammatory. I talked already about engineering, and here this is just kind of a picture of like showing on how many levels you can manipulate your exosomes. So you can fill it with mRNA, currently very interesting, microRNA, long coding RNA, circular RNA. You can fill them with proteins, and you can change the lipid composition. You can add you can add receptors. So all of these are examples that are already existing on like the preclinical level, but I haven't seen anything of this yet, translation into kind of human work. And again, the slide where like how can you help AI, just do more of it, because yes, with kind of like harvesting this from the blood and doing multi-omics clocks, we also need to look at the subspecies of the exosomes and have to understand what's in them and how much they are contributing to your clocks. Better software, better devices, more chemistry, because they're fantastic. You can do loads of interesting stuff with drugs and filling them. But we also have to do more on like the education and teaching side, because I think there is a lot of confusion, and the International Society for Extracellular Vesicles is trying to do that, but basically they need more. And then, as usual, data visualization and data analysis is always welcome. And I think that's the end. So, questions?
Yeah, good question. The bike up.
That takes Nguyen's site where your audio presentation, some of the positive exosomes that we use break out for us to extend our longevity lifespan. Right.
Can you repeat the first bit?
Sure. So you cite earlier in your presentation any specific examples of exosomes where you can actually that well for us to apply for our own longevity or lifespan?
Legally, no. Unless you're part of a clinical trial, there is no way of getting them. Because, as I said, they are not yet an FDA-approved product. What people are doing in other countries, yes, I can't comment on that one. So, as a normal consumer, nothing is available yet. You could do exercise. It's known that exercise produces more beneficial exosomes. As usual, exercise is always good. Having a good, healthy diet seems to be correlated with vesicles that are more good for you. But yeah, beyond that.
So I turned chronologically to 15 years old. The biological would be just 21 years old. And so, to your point, I'm incorporating these the standard health and wellness therapies and the protocols as I might still, but also going to other countries like Colombia, Panama, where we can access kind of the cutting advantage of what's a trouble and leverage for us. So those two existent. I don't think it should matter whether or not the FDA has approved something. Makes sure there's an opportunity for us to surface, and besides, what's working with people willing to apply these therapies to themselves. And so I love to see more and frictionally if anybody comes to ask me what's about the pattern. Let's say package.
Yeah. So in relation to that, yes, you can use your own exosomes in other countries, but I'm just saying again, caution. You might have cancer and you don't know it. You might just kind of like give yourself a shock by accumulating more of it and then doing an injection. You might have some inflammation. Again, like it's probably not good to do that just blindly.
Yes. Test and myth.
Yes, so I would just love that the clinics that are currently doing exosome therapies would do a little bit more on like analyzing what they're actually isolating and how good that is for like a person to get back.
Fun.
Okay, now
So, can you say more about the AI?
it's basically very similar to like the earlier talks on the clocks. So mostly there are statistical models that are being used to look like what kind of RNA is present and what kind of signatures do young and positive exosomes have versus exosomes that come from elderly people. So this kind of like thing, I haven't seen anything on like modeling or so. So mostly it's statistical analysis that is being done.
Then is there feels like excessive weight where like how easy is it to like actually squeeze for what's inside? How can calculate?
Very much in the same way as you would go about like doing single cell sequencing. So you would isolate the exosome and then you could do sequencing. You could look like what kind of DNA or RNA is in it. And then you could also do proteomic on them. Basically, same thing as what you would do with a cell. You do a proteomic analysis, then you understand what kind of proteins are in there. So it's very similar to all of your cell studies. It's just that they have only been discovered very recently. So we are not yet at the same level as like we are currently with cell analysis. Yes.
Hi. I have a friend of mine who was a GMP lab in Singapore. I was trying to do like Mr. Fanny stem cell therapy for elderly with cartilage defects or what is called osteoarthritis and age. And specifically, I think right now there's even some drugs of holidays to prevent the progressive they need. We're trying to do allogenic. So I think the long diagram case is really interesting. What they should do via eye, which is like that's where somebody is in there. And so I'm just wondering, like, how do you see AI can play this? Because I think New Limit is trying to do something similar. They're trying to apply some AI, something even like some physical, mesh together. Wondering if you've seen any companies out there that's doing something like that.
Okay, so how could AI, for example, help make better cartilage? So like MSCs need to differentiate into cartilage. And unfortunately, what you need is you need to have the right kind of mechanical stress for them to really make good cartilage. So people are trying to mimic that in ex vivo, basically in bioreactors. So they apply pressure and they kind of like play with like, okay, how long do I have to actually apply this pressure? And I think this would be an area where like modeling would be relatively easy because it's only a couple of like elements that needs to be shifted. And it would save a lot of time playing with this as we currently do, kind of like, okay, today 5 bar, tomorrow like 10 bar. If I would have a model that would help, that would really help also companies making better cartilage. So just one example of how that could be integrated, yeah.
Sorry, just to clarify, we talk about MSCs and coordinator cells, right? And it stress within the scaffolding so that it improves the holding ability. Is that what you're saying? Because I just want to make sure that I cover price.
Well, it depends. Okay, so scaffolding can be taken into the process if you want to make cartilage. So you can provide the MSC some scaffold that helps them make good cartilage. But you could also think about ways of actually transplanting a scaffold in the body to create kind of a niche for your stem cells that are already in your body to make better cartilage. So I've seen people playing with both things. It's possible. On the question, are MSCs progenitors? The whole MSC field is always fighting if that thing is actually a stem cell or not. So I won't kind of like comment on it. Just read the literature, make up your own mind.
Sure. Do these with exos stay away with anticipatory and outside?
So no, the exosomes actually travel via the bloodstream. So if you would inject something in your left leg, you would see them also appearing in your right leg. So basically, they also appear in your brain. So they also travel across the blood-brain barrier. So they seem to be relatively stable and able to move around in the body.
So what was it anything to like carbon assist best?
Could be. I have not yet seen a pyrolysis experiment that looks specifically at the role of exosomes, but it's theoretically possible. Good. Looks like everyone is fine. Good. Thank you very much then.
Okay. Thank you, Aubrey and Alex. That is perfect because I was told.